So I just want to talk today, for completion's sake, about a couple of concepts regarding variation on Mendelian inheritance. So we are all now very familiar with Mendelian inheritance, but in biology, it's typically the case that the rules are always bent. There are always minor modifications. And so although the rules and the principles provide guidance in understanding basic observations, what we always want to be aware of in biology is that typically there's very frequently there are variants on those principles. And that's what we'll talk about today when we talk about the concept of incomplete dominance, codominance, and then I'm going to define for you expressivity and penetrance, and use that to introduce a couple of useful concepts thinking about relative risk. And then we're going to switch gears, and this is really a transition in the class where we're shifting from transmission genetics, that is understanding the transmission of alleles in families, to population genetics, and that's thinking about alleles in populations and the forces that act on them. We'll be introducing that in the next few lectures. So let's just go back now to Mendelian inheritance and the concept of incomplete dominance. And so here what we're looking at is are some beautiful flowers. And we have, in this case, a red flower, an ivory flower, and they breed true. But when we cross those flowers, we don't get a red flower or a white flower back, we get a pink flower back, okay? And this, one might think, is, in conflict with our understanding of Mendelian inheritance. But in fact, if we introduce the concept of incomplete dominance, then that is that our big R allele doesn't act with complete dominance, but actually is incomplete in its effect, then we can, then this scenario of blending of traits is consistent with Mendelian inheritance. And so we can demonstrate this by taking a parental cross and crossing the red and ivory plant flowers and getting our pink F1. But then if we self that F1, we get back in the F2 generation, both red and ivory flowers in addition to the pink flowers. And if we count the numbers and look at the proportions, we get them in the expected proportions consistent with Mendelian transmission genetics, okay? And so this is a really important concept, and it's interesting to think about the molecular basis of this. So in this case, there's an enzyme that produces the red pigment, the R enzyme, but it has a dose effect. So if you only have half the dose, then you get half the amount of red pigment, and so you get this blended phenotype is what's manifest, okay? And so this is the phenomenon of incomplete dominance. For humans, an informative case to look at is ABO blood groups and how that affects, or how we can think about inheritance and variance on Mendelian inheritance with respect to the ABO blood group. So does everyone know what blood type they are here in this class with respect to the ABO locus? That's good information to have. Is anyone here a universal donor? I'd like to know who the universal donors are, just in case. If maybe you don't want to tell anyone because your blood is very precious, so it's probably a good idea to just keep that on the low down. But universal donors, of course, can donate to anybody, and universal acceptors can receive blood from anybody, but knowing your blood type is very important for blood transfusions. And so you can either be an A blood type, a B blood type, AB, or you can be the O blood group. But your blood type is with respect to the ABO blood group type. So C told us that they are A plus. I think the plus refers to the rhesus locus, which is another locus and another epitope or antigen that's expressed on the on red blood cells. But we're focusing here just on the ABO blood group. It's a single gene that's called the ABO gene, but there are 3 common alleles in the human population. And these we'll call the IA allele, the IB allele, and the IO allele. And so depending on which of allele you have, which allelic variant you have at the ABO gene, this determines your phenotype in the following way. So the key thing to know is that this locus encodes a glycosyl transferase, okay? And so let's look here at the carbohydrates that are on the red blood cell. And so here's our red blood cell. And if you're type O, you have the following carbohydrate structure on your blood cells, okay? And this is an epitope or an antigen that's stuck on the blood cells. And if you are type A, that means that your allele at the ABO locus is the A allele, which encodes the A transferase, and that attaches an N-acetylgalactus. Who can pronounce this for me? I'm not able to pronounce it right now. Acetylgalactosamine. That does not sound right.
It is.
Can you just say it so I can hear it?
Acetylgalactosamine.
Acetylgalactosamine. That sounds a bit better. Yeah, I think I was putting the accent on the wrong syllable. So when you have the A allele at the ABO locus, it will attach this molecule to this backbone. This is the backbone that is common. This is the one that's in the O blood type here, and it attaches this molecule. By contrast, if you have the B allele, then you attach a galactose. That one I can say easily. Okay, and so now on a B, if you have type B blood, then you have the galactose attached to, again, this backbone. That is the backbone that is found in all individuals and is found in type O. Okay, so this is how the ABO blood groups work. So you have type O people do not have a functional transferase. Okay, and so since they don't have, since they have essentially a null mutation, they lack the ability to attach an additional molecule to this carbohydrate backbone. And so this results in the absence of any additional sugar being attached here. All right, and here I put a little blurb about that, about what the actual molecular characterization of the ABO locus. It's on chromosome 9. You're all now comfortable with the nomenclature for chromosome 9q. Q is the large arm. Remember, P is the small arm for the French petite. Q being the large arm. And so this gene, the ABO locus, is on chromosome 9, 7 exons, 18 kilobases of DNA. Exon 7 is the largest. Exon 6 contains the deletion that is found in most O alleles and results in the loss of enzymatic activity. So we call this a null mutation. So if you have the O allele, you essentially have a mutant form or a non-functional form of the gene. And the A and B alleles differ from each other just by 7 nucleotide substitutions or 4 amino acid differences. And these are the amino acid differences that are shown here. Does everyone know how to read these? Read this? No? Okay. This, who knows what this number is right here? If we're talking about an R176G mutation, that is the amino acid number or the codon. It's the 176th codon, okay? And how many nucleotides comprise a codon? 3, good. And so you should be able to tell me what nucleotide position this is in the coding sequence then, right? What nucleotide position is this codon at? Okay, 1 codon, 426, 526, 528. How'd you do that? You did 176 times 3, right? What is 176 times 3? 528. Okay, so that means that the first nucleotide of the codon would be at position 528. Does 3 go into 528? 3, that's 1, 4. Yeah, good. Okay, so that's the codon where the mutation is. Here's the next codon, here's the next codon, here's the next codon. And what this nomenclature, this way of writing mutations tells us is that in the wild type, there is an R encoded by that codon. What is an R? Does anyone know? Arginine, right? It's like the pirates. What's a pirate's favorite amino acid? Arginine. No one's heard that joke before. I'm sorry, that was pretty bad. Okay, so in the wild type, there's an arginine at position 176, but it's mutated in this allele to a G. What is a G? Right, so it's an arginine to glycine mutation, okay? And so you should be able to know, this is why Molex was important, right? You memorized all 20 amino acid symbols, UPAC symbols, right? I could put that on the midterm next week and we'd be good. No, okay, it's a good life skill to have. Biochem, yeah. Anyway, you should aspire to do that. You should aspire to know, so here, these are all, what would it S be? Does anyone know? Good guess, serine. L, leucine. That's good because it's not lysine. What is lysine? Okay, nice. M, methionine, and A. All right, I'm impressed. You've got it. What about what's this one? Y, tyrosine, tryptophan. Then the ones I get stuck with is this one's a little hard. What's that one? D, aspartic acid, and oh, here's a good one. What's this one? Q. It's my favorite amino acid. This is my favorite amino acid, glutamine, right? Okay, so this is writing this nomenclature for indicating these mutations. So these are coding mutations. That means they are non-synonymous mutations, right? That's why we have 7 nucleotides, but only 4 of them translate into different amino acids because you can have non-synonymous mutations and synonymous mutations, which is where a mutation still results in the same codon, and because of the redundancy of the genetic code, okay? And if we read on, we see that it's only position 266 and 288 that determine whether you are A or B. So that's pretty amazing. There's only 2 amino acid differences that determine whether you're an A blood type or a B blood type. And that's because it determines, the enzymatic activity of the glycosyl transferase. There's also the O allele differs from the A allele by deletion of a guanine at position 261, so just one base, but deletions, as we recall, can often call frame shifts, and so the rest of the amino acid sequence is scrambled with respect to the wild type, and so you get this junky protein that doesn't do anything, okay? And that's the mutant type. Okay, so now you know all that you need to know about the ABO blood groups. All right, so what's interesting about, and this is unusual actually in humans, is that we make antibodies to the opposite antigen to which we have, okay? And this is why you need to know your blood group, because if you are blood group A, you have the A antigen on your blood cells, but you make anti-B. I don't know why the human body decides to do that. I'm sure people understand, hematologists understand this now, but this is all you need to know, is that you make antibodies to the opposite antigen. So if you are group AA blood type, then you make anti-B. If you are BB, so you have 2 copies of the B allele, you have the B antigen and you make anti-A. But if you make both the B and the A, because you are an AB blood group, then you do not make any antibodies and you are a universal acceptor, okay? You can take anybody's blood because you're not going to mount an immune response to somebody else's if you get a group A blood because you don't have the antibody to it, okay? By contrast, if you're group O, you don't make any of these antigens, but then you make anti-A and anti-B. These people definitely need to know their blood type because they cannot take blood transfusions from anybody except people who have O blood type, group O blood type. So you have to be O homozygous. So the genotype actually has to be, you have to have both copies of the non-functional gene, which encodes the non-functional transferase in order to be O blood type, okay? And this, from a molecular perspective, so when we talk about a phenotype, we talk about, height, eye color, body weight, that sort of thing, but we can also talk about molecular phenotype. So with respect to the molecular phenotype of which antigen you express, we call this codominance, in which both forms are expressed, okay? And so this is another modification of our ideas of dominance and recessiveness. We can have codominance in which two versions of a phenotype are expressed. And so what I want to make sure that you leave with the impression of is that when we talk about dominance, dominance is a property of a pair of alleles in relation to a particular attribute of a phenotype, okay? So a useful example is the condition sickle cell anemia. Sickle cell anemia is caused by a mutation in the hemoglobin gene, and it results in this sickling phenotype in hemoglobin cells, in red blood cells that have this mutant form of hemoglobin, okay? And so we can look at either the molecular phenotype, that is the molecular structure of hemoglobin. We can look at the cellular phenotype, that is whether the cells undergo this sickling or this sickle shape or a normal morphology. And we can consider the phenotype of anemia, which is that individuals who have these sickle-shaped cells have reduced capacity to transport oxygen and have iron deficiencies, and so have the condition of anemia. Okay. And so with respect to these different phenotypes, either a clinical manifestation, a cellular morphology, or a molecular phenotype, they can vary with respect to these alleles. So with respect to the normal wild type, the A allele of hemoglobin, those individuals who are homozygous for wild type do not have anemia, do not manifest the sickling cell morphology, and have the A form of the hemoglobin. But those individuals who are homozygous for the mutation in the hemoglobin that causes this disease have severe anemia and have a high sickling phenotype, and they have two mutant copies of the protein. But those individuals who are heterozygous do not have anemia, but they do manifest some sickling. Okay, and they have both the mutant and the wild type form of the hemoglobin. And so with respect to this one mutation, this makes the point that when we talk about dominance, it's a property of the pair of alleles with respect to a particular attribute. Okay, so we can, with respect to anemia, we see complete dominance. So individuals who are heterozygous for the mutation do not manifest anemia, so you have to be homozygous. Oh, sorry, this is dominance with respect to the wild type allele, right? The wild type allele is dominant in this case. So if you have the wild type allele, you do not manifest this. In this case, because we have an intermediate phenotype, we call this incomplete dominance. And in this case, because we have both molecular phenotypes, both the mutant and the wild type form of hemoglobin are expressed, just like with the AB blood type, we refer to this as codominance, okay? And then finally, just thinking about variations on the Mendelian inheritance, there's two important concepts that we need to know about. And these are particularly important with respect to human genetics. And that is that the expression of a mutant gene can differ between individuals. So in two different ways. So we can either have variable expressivity, which refers to traits that are expressed to different degrees in individuals, or we can have variable penetrance, which refers to the proportion of organisms, or humans in this case, whose phenotype matches their genotype for a given trait. And so we can talk about variable expressivity and variable penetrance, the two important concepts. And so here's a schematic to be able to understand that concept. So variable penetrance means that if we have a mutation, a dominant mutation that causes the phenotype purple, but in some individuals, they have that mutation, but they are not purple, they maintain the wild type phenotype. And so if we look across many individuals, we see some manifest the phenotype and some do not. This is a scenario known as variable penetrance. There's incomplete penetrance of this mutation. This is in contrast to variable expressivity, where all the individuals manifest the phenotype, but they manifest it to differing degrees. And so there's variation in the expression of the phenotype. And then to further complicate it, we can have combinations of variable penetrance, where only some individuals have the phenotype. And among those individuals who have the phenotype, there's variation in the expression of that. Okay, and there's two well-known examples of this. So retinoblastoma is a congenital cancer that occurs in the eye due to a mutation in the RB gene, which is an important cell cycle regulator. But you can have kids in the same family who have inherited the disease allele, and some of those kids will have the cancer and some of them will not. And so this is a scenario of variable penetrance. The mutation is the causative factor, but there are individuals who have the mutant allele who do not have the cancer. By contrast, neurofibromatosis is another source type of cancer that occurs in the nervous system, and it's manifest by these splotches on people's skins, and called cafe au lait splotches is the diagnosis, is the clinical presentation of this. And so you can have individuals who have that mutation but have very different extents of this clinical manifestation of these different cafe au lait spots across their body. And that's a case of variable expressivity. Even though all the individuals have the same causative mutation, the extent to which it is expressed at the phenotypic level varies between those individuals. Okay, and so penetrance is actually a good case of thinking about conditional probability, because we can think of penetrance as the probability of expressing the phenotype given your genotype, given that you have a mutation. So the probability of having retinoblastoma given that you have the retinoblastoma gene. And this is actually useful for being able to quantify how your increased risk with respect to having that particular gene. So let me just, I've got one thing I want to show you on my screen. Okay, so as I said, penetrance we can think of as the conditional probability of some phenotype given some genotype G, okay? And we can think about simple cases. So for example, the probability of having the B blood type, given that you have this genotype with the, where you're heterozygote, is going to be equal to 1 because the B allele is dominant in this case, okay? Whereas we can think about the probability of having the O blood type, given that you have that same genotype, that's going to be equal to 0, okay? And in fact, dominance is distinguished by the penetrance of heterozygotes is one way to think about it. So dominance is distinguished by the penetrance, the probability that the phenotype is expressed given your genotype of heterozygotes. Because in this case, this is dominance, complete dominance, and in this case, this is complete recessiveness. But there are diseases for which the probability of having the disease given some genotype is less than 1. Okay, it's not equal to 1. It's incompletely penetrant. Okay, and this is because of this variable penetrance, or because of variable penetrance, or we also say incomplete penetrance. And so we can define a metric that's often useful, which is that we can say, well, what's the probability of being affected given that you have some mutant genotype, some disease-causing mutation, for a disease for which there are other causes, okay? So for example, human cancers are a great example of this. So an example that we'll look at is the BRCA1 mutation. BRCA1 mutations dramatically increase your risk for breast cancer, but there are other mutations and other causes of breast cancer. So we can think about those as geneticists as being phenocopies. So we get the same phenotype, but for a different underlying genetic or environmental reason. And so we can think about, well, in that case, we can think about the probability of being affected given that you don't have the mutation. We'll or don't have the mutant gene. And we define this value as a penetrance ratio. This is a useful metric for being able to talk about how the relative risk of having that mutation, okay? And so this is actually in epidemiology, this is defined as a relative risk. And so let's think about an example of the example of breast cancer. We can think about the probability of getting breast cancer given a BRCA1 mutation, given that you have inherited a BRCA1 mutation, and that number is about 0.6. So you're at a very high risk of about 60% chance of getting breast cancer if you inherit the BRCA1 mutation. But there are other sources of breast cancer, and so we can think about the probability of getting breast cancer in individuals given that you have a wild type, BRCA1, and that's about 12% of women, okay? And so we can think of the penetrance ratio or the relative risk as being simply the ratio of these two, which is 0.6 to 0.12, which equals 5. And so therefore, we say that you are 5 times more likely to get breast cancer if you inherit the BRCA1 mutation compared to the general population, okay? And the reason why, so this same concept is used, for example, you can, it's exactly the same epidemiological concept as saying, e.g. for smoking. So in the olden days, people used to smoke these things called cigarettes, and they would increase your risk for lung cancer. So again, you can think of, the probability of getting lung cancer given that you're a smoker divided by the probability of lung cancer given that you're not a smoker to define the relative risk or your increased proportional risk given some risk factor is exactly the same idea. Those are our variants that we need to be aware of: codominance, incomplete dominance, variable expressivity, and variable penetrance or incomplete penetrance. Okay. With that, we are ending our journey through Mendelian inheritance in humans, and we'll be switching to population genetics.